me introduce you, Dr. Sacco. Dr. Sacco is the president and founder of Alleato Consulting. She has 20 plus years in this industry, small and large companies. In fact, she's been instrumental in the development programs of quite a variety of drugs that I've personally prescribed: Actonel, Lyrica, Rezurim, Actos, you name it, and some more recent ones as well. No, it's an honor really to have someone from your stature and background in industry present on clinical pharmacology and the benefits and challenges in clinical development. So without further ado, the floor is yours, Nancy.
Thank you so much. I appreciate the introduction. I hope I can do justice to the topic since someone else was going to be talking about this, and I'm filling in and hope that they do feel better soon. I do want to bring up the fact that when you're talking about clinical pharmacology, we could spend an entire week talking about how clinical pharmacology is involved in drug development. And most of us hear it and see it as phase one. And when you look at the paradigm of developing drugs that I'm sharing here, we know that we go from the bench to non-clinical models, and once the FDA has accepted an IND, we go into those first-in-human studies. And we tend to think about the first-in-human studies as the clinical pharmacology, clinical PK study. But when you really look at drug development, clinical pharmacology data is really collected throughout the development of any particular new drug or therapeutic intervention. The phase one studies are really, if you look at how the FDA defines it, it's really to determine safety and tolerability of that new drug. And that's how we look at the phase one, first in human, healthy volunteer studies. But throughout the course of the clinical studies, whether proof of concept, phase 2B, phase 3, and now even the phase 4, as many drugs change formulations over their life cycle, there is additional clinical pharmacology data being collected. And it really is tied to strategic planning of what type of data or clinical pharmacology study one does and when. There are very clear expectations that both the FDA and EMA and PMDA do expect, for example, of this kind of data, which is looking at how the drug works in humans. So you really can define it as it's making use of the data that allows a physician to properly use that drug for optimal effectiveness with the least negative effects or adverse effects. So we designed the studies for the purposes of human use, be it healthy volunteers initially and then into the target population. As I mentioned, we find this data useful through all phases of the clinical trials. But the key to remember is that this is very important scientific and analytically data-based decision-making that we do with this particular data set. We look, for example, on the more specific clinical evidence. If it's a pain medication, is pain reduced? If it is a sleep medication, is sleep now improved? Those types of patient-reported and clinician-assessed parameters are the things we think of most obviously. But the clinical pharmacology data looks at how the drug works within the body, how it's absorbed, how it's distributed, how it's metabolized, how it's excreted, and gives us some very important information that then is translated into a number of different ways by which the physician will make determinations on use of the drug. I thought it was interesting that if you go back and you look at some of the clinical pharmacology and toxicology articles and you look at some of the literature behind all of this, they do at times refer to it as an ancient science. And it was interesting to see them refer to things such as, excuse me, hemlock. We're going all the way back in centuries. It was really looking at how an herbal medicine um, affected the body. And that's the simplicity, perhaps, of how you are looking at how a drug will work. But I tend to use the more beneficial one as an example, and that is insulin. The use of insulin relative to glucose levels. And again, that is the clinical pharmacology behind it. But we also see very many different terms. It's not unusual to see pharmacokinetics, pharmacodynamics, pharmacometrics used somewhat interchangeably. We know now that we're looking very closely at pharmacogenomics, the pharmacotoxicology data. I've already mentioned metabolism and clearance, and truly to look for optimal dosing and dosing regimens. And so what we're really talking about is we take data from the non-clinical studies and we look at them very carefully relative to a no observed adverse event level, or NOEL, or we might look at them in terms of a minimum anticipated biological effect level, or MABEL. And that's how we begin to think about what is the dose or dose ranges we might look at in the first in human studies and then in a number of other types of studies. So what I'm going to share here is just an example of some of the different types of clinical pharmacology studies that are needing to be designed and operationalized depending on the class of the compound. The first in human I've already mentioned, most often in healthy volunteers, but there are those indications where we don't go into first in human with a healthy volunteer population. In oncology, for example, we will assess the doses, the safety, the tolerability of a new drug in the oncology patients themselves. It's a smaller study, sometimes as small as 12 to 30 some healthy volunteers, depending on the dose ranging. But if you're talking about, for example, a biosimilars program, your first in human PK/PD studies, which are really in terms of a reference product, will be in this area of 100 to 200 some volunteers. We will then look, for example, you can take your first in human and design it to be a true PK/PD type of study where we're looking at single dose and multiple dose rising concentrations or doses of the drug, which we then will refer to as the SAD/MAD type studies. And they are really looking at the difference of the concentration of the drug that is being given over time, as well as perhaps looking at that concentration relative to a biomarker. So we thus refer to them as PK/PD profiling studies. I've already mentioned the ADME for absorption, distribution, metabolism, excretion, oftentimes done in non-clinical studies, but that becomes very important in really assessing the profile of that drug. As you go on in developing the particular drug, you do start to think about special population type studies. Those could be renal or hepatic impairment. They could be looking at how the drug works in an elderly population versus a pediatric population. If you just saw the news yesterday, for example, the FDA did give approval for the COVID-19 vaccine to be used in an adolescent population. And you can, if you have been following it, you see how the information around the drug or the vaccine in this case and the data really looked at the population in an age-related and risk-related manner. And the more information they have, the more ability to expand the use of a particular drug. We would look at drugs, for example, relative to gender and race. And I know that for those of us who have been doing this for quite a while, when we look at this, we realize some of us have actually been seeing the change in how first-in-human studies used to be referred to as first-in-man studies. But we know that many of the diseases we are developing treatments for, or have been developing treatments for, are not only a disease that impacts men, for example. And so we have seen them turn into studies as looking at both impact and effect in men and women. And in race, there are differences. In clearance, simply if one looks at liver metabolism, the P450 aspect, and one sees differences in clearance rates of drugs. So you may have a ethnobridging study, which looks at a predominantly, let's say, Caucasian cohort versus a cohort that is looking at the clearance race in Japanese. And that becomes very important when you are looking at global use and you are looking at a heterogeneous population that really suffers from the same disease, and you would like to be sure that the drug is being used properly regardless. We also have, for example, when we're talking about vaccines or monoclonal antibody treatments, these also are drugs or therapeutic interventions that can result in an immunogenic effect, an adverse effect, or simply an allergic response in a simplistic manner. So you begin to look at immunogenicity. And it could be related to gender, it could be related to race, or it could simply be related to the class of compound itself. When you're looking at diseases, chronic diseases especially, in an aging population, you begin to realize that there are multiple drugs that someone may be taking, and any new drug that might also be used in this population, we do need to have a sense of how does drug A impact or interact with drug B, because we don't want there to be any increase in safety issues or adverse events, and we don't want one drug to cancel out the beneficial effects of the other drug. So it is now looked at in terms of a key piece of information in the course of developing new drugs to see how various drugs will interact with each other. There's also a continued element of safety. It's one thing to collect safety data in healthy volunteers. It's another thing to collect safety and monitor safety in patients who are already suffering from a disease or maybe immunocompromised, for example, in the oncology patients. And I'm providing here an example of the cardiovascular safety. There are those of us who, in the process of development, have learned that the last thing you want on a drug label is to have a black box. And that is truly the notification of there is some severe or high risk adverse effect of this drug, and one needs to know what it is in order to use it properly in patients. And so on the aspect of cardiovascular safety, sometimes, depending on the class of drug, let's say the quinolones or certain antibiotics, one has to do a thorough QTC study because prolonged QTC is truly detrimental. If it continues, you end up with torsades. and you really end up having very severe issues with the drug and potentially a drug that could not be approved. There's bioavailability and bioequivalence if you are looking at comparability between generics and, as I've already mentioned, biosimilars. And there are also food effect studies. There are certain drugs where the absorption and metabolism, even the clearance, will be quite different if they are taken with food versus without food. So you can see from this list, when we're talking about clinical pharmacology, it really isn't just that phase 1, first in human or SAD/MAD type of study. It is really looking at how the drug, the concentrations, the doses, the dosing regimen, proceeds over time in certain and specific situations. And so that's why it's really looking at how the drug impacts the body and vice versa. So I'm going to take that original FDA paradigm and use what I think is one of their best slides in looking at the clinical pharmacology studies over the course of development. It's not unusual at the end of the development of a drug to find that you have probably about 6 or so clinical pharmacology studies specifically as their own, but you will have PK/PD data in all phases. And you see here that they now do have phase 4 within this paradigm because drugs tend to change their formulation in some way, or perhaps even the delivery system. So this gives you a sense of how, from the time of the preclinical work all the way through to approval, where and the different types of pharmacology data that one is collecting, either as individual studies, as I mentioned, the SAD/MAD, a food effects study. We might be looking at PK and renal impairment, hepatic impairment, and now we do need to have some specific data of PK in pediatric populations or geriatric populations, or certainly in women of childbearing potential. So this is all very important. And when you are designing the studies, I think this is the area of clinical trials right now that has quite a bit of focus, especially as this COVID period and post-COVID period has moved us more and more toward decentralized trials. So I wanted to bring up a couple of points. When you're doing the phase 1 clinical pharmacology work, we really are talking about building out data from intensive sampling. And those of us who have done these studies and have the phase 1 studies in a unit, a phase 1 unit, we know that they can start collecting samples within minutes of dosing and continue over a 24, 48-hour, or maybe even 7 or 8 day period. Think about that relative to this movement into decentralized trials, which is very important. But when you're doing this first building out of population PK or information about how the drug is working, having the ability to collect those samples in this time-oriented and focused manner is extremely important, especially when we start moving into the phase 2, phase 3 studies, where the decentralized trial model is certainly moving toward. And so now we know for sure we're going to have far less sampling or ability to sample because we were leaning on, as an industry, to have those samples collected whenever the patient came to the clinic. If we are decreasing those visits, we now have a different operational situation to consider that impacts the outcome and the science behind the use of the drug. What we're talking about now is when we have the telemedicine type checks or the technology of transfer of data, be it vital signs or other data, electronically back to the site or the CRO managing the study, that's not the way to collect the PK sample. We can't do that. We might be collecting the PK sample by blood, serum, plasma, urine. Perhaps in the oncology patient, it's not unusual to be taking some tissue to look at concentration. So how important does it become to have the earlier data? It is even more important now. So the preclinical studies really do have to have much more robust PK information. The phase 1 study and the clinics have really moved toward assuring us as sponsors that the patient, the healthy volunteer in these studies or the oncology patient are well treated, that they are safe, that the staff is trained on all of the CDC and PPE requirements so that we can still get the intense sampling that we need, because that now becomes very critical for predicting and modeling when and how we want to collect PK/PD samples and assess endpoints in the patient trials in phase 2, phase 3. When you are talking about special populations, we are again fortunate that many of the units that have restructured a bit how they operate can also continue to have the hepatic and renal studies done there in very much the same way and protected manner, and also drug-drug interaction studies. The food effect studies, just from most recent experience, are becoming a bit more hybrid. The sampling is still intense, but not as much as in the first in humans SAD/MAD type studies. But you can now, there are CROs who not only will have the visiting nurses go to the patient or the volunteer's home, but phlebotomists are now in some cases being used to go and to collect the samples. The less processing the sample needs operationally, the easier it is to use the decentralized approach with that type of clinical pharmacology study. So when we're looking at the science behind the study, we know those clinical endpoints, either from discussions with the regulatory agency or knowing the target or knowing the general outcome. Again, I'll come back to pain, which is one of the more obvious ways to assess effectiveness of the drug. So the design will look at those primary clinical outpoints, but you will also have to be looking at the timing and what samples are needed for the PK/PD. So that has to be built into the study design effectively and feasibly right now. Behind that, we also need to be sure that the methods that are being used to evaluate the drug concentration are methods that have been validated. You don't want to be changing the method of evaluating drug concentration over the course of development. You really do need to have, and the FDA has some excellent guidelines out there on the expectation for method validation for the purposes of PK and PD, starting with the first in human study. This also means that sometimes we need to consider specialty lab capabilities. There are certainly different platforms that need to be used to do some of the drug concentration assessments. It might be HPLC, liquid chromatography, it might be Luminex platforms, it might be ELISA. Not all of the central labs have these specific methods available. They don't necessarily use them on a daily basis. So you may have to go to a specialty lab. that really focuses on PK/PD, methods development, methods validation, and assays specifically that can be used and will be up and running in time for the first in human and all the way through the course of the development of the particular compound or drug. The collection methods I've already mentioned. As we go into some of the decentralized trials or siteless trials, as we talk about them, depending on what's being collected, a nurse may be able to obtain the sample and easily get it prepped, packaged, and shipped either to the lab or to a site or wherever the assay is being done. And the less we have to prepare that sample, the better, but that's not always the case. Many of the PK samples do require tissue homogenization or may require serum versus plasma versus whole blood. Obviously, the easiest medium for the patient or volunteer at home to collect is urine, and they may have nothing more than instructions such as placing the sample in an amber colored bottle, if it's something we're worried about light sensitivity, or place it on ice, or simply collect over a 24-hour period, and it'll be picked up when a call is made. So it's important to know from earlier non-clinical data and non-clinical studies what's the best medium in which we can test the drug concentration. We've already talked about the collection times. The storage, I've already mentioned, something may need to be placed on ice, may even need to be frozen. So some of these operational aspects will impact the ease of doing decentralized trials or how we can distribute the collection times so that we can still offer the patients who are enrolled fewer visits to a site, to the clinic, but still collect the data that we need. The sites may still have to be shipping to the specialty labs, as I've mentioned, but I'm going to bring up another piece around the operational challenge. The analytical plans need to be very clear. It's possibly you may be thinking about how to analyze those samples as they come in. In many cases, for the purposes of PK/PD, we have worked more closely to analyzing them in batches, perhaps by cohorts, perhaps by a time point, and how quickly the analysis will be done once the last samples have reached to the lab. And that point, the data management and transfer is usually done to the CRO, can be to the CRO who is managing the study for very specific statistical analysis and modeling to be done, or it might go to a niche vendor or consultant who takes that data and really puts it through the modeling approach and very specific analytical assessments. A lot of this I've already spoken about, but I just want to come back just to recap a few of these things. The plans for the methods development and validation, we as sponsors have very specific guidances and requirements that we know the agencies expect. They expect certain variability parameters, and if it's outside that, they do not consider the data robust enough. They expect clarity around the methodology and the reproducibility of results. So it is a very, very strict set of bioanalytical requirements that we meet, and it may be different, as I mentioned, depending on the types of platforms that we are talking about. You may have to have binding antibodies. If we're talking about a monoclonal antibody development, there's additional PK/PD assessments that are done. We're looking not only at the monoclonal antibody that's been dosed, but we also then have to assess whether or not the patients begin to develop antibodies to block the monoclonal antibody and inhibit its effects, or neutralizing antibodies as well. So we really have to look at what's being developed, the class, and what is expected based on what we know now over years of different types of drugs being developed. What else do we look at? We may also be looking at some very specific biomarkers. And you do like to have those assays oftentimes done in the same lab that is doing the bioanalytical work for the drug concentration, so that they are looking at assessments in real time or in parallel, and all of that data is kept together and then analyzed. We do need to be sure that the specialty labs are able to work with the clinical sites, providing them instruction, materials, whatever is needed, and that they also work very closely with the CROs who are managing the clinical trials. Everyone needs to understand this is a very finely tuned machine, and as we move to decentralized approach, the oversight of how some of these special samples are being collected and how the data is transferred becomes even more important. And I put here, I highlighted the location of the CRO and the bioanalytical lab. Couple of things to keep in mind. We now tend to do these global trials, and we have all lived through the fact that biological samples are not normally allowed to be moved around the world with great ease. There's some very strict requirements. And when you're talking about things like the PK sampling, it's always good to be working with a lab that can have that technology around the globe. So if you cannot move samples out of China, you have a bioanalytical lab in China that can take that same technology and platform and run those samples there. And then that data is merged with the sample data, the analysis you have from sites in Europe and from sites in the US. So you have to be very clear on how and where these samples are going to be stored and handled. I've already mentioned the basic PK/PD, but there's different ways of analyzing the data, compartmental and non-compartmentalized analysis, whether you're looking at specific tissues, specific targets. And then there are the key parameters along which we determine the drug absorption, metabolism, and distribution. Are we looking for the half-life? Do we want to know the maximum concentration? Are we looking at the area under the curve in order to assess all of that? And I'll go through that just very quickly. And this is why I'm saying, from an operational and technical standpoint, we have to be very good at making sure whoever is handling this data and doing the analysis knows what they're doing and how to analyze that data. Smaller companies tend not to have the core group or core pharmacology group that will do that type of analysis, because that really is a very specialized science, so to speak. And so they may go to niche CROs and statistical analysts to do that work. And I'm just showing this as an example. For the PK, as I mentioned, area under the curve, the Cmax, the Tmax, the half-life of a drug, all of these aspects allow us as the sponsors to really determine what is the best way to dose the patient. Where do we get the maximum effect and at what dose with having the least amount of toxicity? So we look at exposure, we look at efficacy, and we look at response and we look at exposure in order to be able to see how those curves balance against each other so that we can pick the best dose for the best effect with the least amount of safety issue. So you tend to link your non-clinical tox studies in looking at the general PK for dose escalation and half-life, as I mentioned. You look at it relative to efficacy and safety biomarkers. You consider the safety margins, which we all know the FDA likes tenfold, but in certain cases, they understand tenfold may not be possible. So there are discussions as what is the lowest safety margin that is acceptable. And over time, with Cmax, area under the curve, accumulation, you begin to develop your dosing regimen relative to the dose. I'm going to come back to the operational piece for a moment. One of the other things that's really important as a sponsor, whether it's with the lab, a specialty lab, and or the clinical CRO, we do have to put data transfer agreements into place. We need to be clear. We need to be clear when they do the analysis, how the data is transferred, how it is set up, what is going to be the time frame, let's say, between the last samples analyzed and the full data set, then moved to wherever the clinical data is being held or managed from in order for the analysis to be done. Again, that does require some understanding of the difference between that clinical CRO that's running and managing the study and these unique vendors that are analyzing some critical data. And then, of course, you begin to look at the different type of analyses. Non-compartmental analysis for exposure in order to make sure we know what dose relative to adverse events. How we will do population or what we expect population PK modeling to be done using phase 2 and phase 3 data, where we're not collecting samples in a matter of minutes, but we might be collecting pre-dose, maybe 24 hours, and then maybe weeks after dosing in order to really do a population PK modeling. You may be looking at those values relative to body weight, age, I've already mentioned ethnicity, or maybe other aspects. And then we do, as I mentioned, we do have to have the population PK now relative to pediatric dosing, especially because the FDA and EMA do expect pediatric plans. And if a waiver isn't going to be granted or requested, you do have to have what the plan is and how you'll assess pediatric doses in a significant amount of time before you do your end of phase 2 meetings. So these all become some of the operational and scientific basis in planning for the clinical pharmacology work. This is also from, if you get a chance, if you haven't ever, go out and look at some of the training information. One of the best training series that I have personally believed the FDA has is around clinical pharmacology. And it is very interesting to see how they basically have created this pyramid, which starts out very broadly, non-clinical to clinical, populations, drug interactions, a series of variables that might impact what the label might be looking like, what we finally put in as the sponsor relative to dosage and administration that we hope the FDA believes the data supports, and any specific highlights that need to be mentioned about dosing and impact of the drug. All of this depends on the collection, the sample analysis, the data analysis, in order for us to really bridge the science with the medical use. And so I wanted to bring this up just to share with you we really, as sponsors, we constantly think about the patient, the outcome, the benefit. And we look at it relative to a primary endpoint about the disease itself. In oncology, it might be morbidity, mortality rates, survival. Again, the simpler diabetes, we're looking at management of glucose levels, the other aspects of indications for pain, the management of that pain. So we think about it just as the patient and the doctor thinks about it. Are you feeling better? Are you seeing benefit in the broadest sense? But when you go and you look at our labels for the drugs, you will see that quite a bit of it is related to the clinical pharmacology. So section 12 of the label, we are looking at mechanism of action, which we oftentimes can tell from some of the PK/PD. We look at pharmacodynamics. There might be the cardiovascular impact, absorption, distribution, elimination, any special population, and drug interaction studies. So it is dependent upon the class of the drug, which of the studies we do, and the samples we collect will depend on the patient as well as the method that is validated for measuring the drug compound itself. We will look at, and obviously now as sponsors, we really are dependent on real-world data and real-world experience, specifically whether it's the patient population and the disease, or we're looking at the drug class. And this is where one of the areas where not only where there's safety information, but this area here in section 12, the more data we have, if we change formulation, it is one of the areas where the label changes will occur. And I just wanted to bring back the fact that clinical pharmacology, from science to operations to technology, is really focused on having the right dose, using it in the right way, at the right time, in the right patient population. And that is really how we provide the benefit for the prescribing physician to understand more about the drug we have developed, how to use it, and how to always be looking at benefit versus risk with the patients they are treating. And so I wanted to just let you know if the slides, Sam, if they're going to be available. I pulled together just a couple of the key guidances around the validation, the data, how they expect data to be presented. And then the ASCPT has a really great summary article on clinical pharmacology and modeling. Thank you.
Excellent. Dr. Sacco, that was a fantastic presentation. So many actionable insights. In fact, I would recommend that presentation for medical curricula throughout the States for sure, and certainly in ClinDev programs as well. No, I will reach out to the Secretariat, the AAMC, because this is the grounding, science. There's so many myths on linear development and clinical development, particularly. You've done all your ClinPharm work. You've introduced very temporal topics about how COVID-19 has created less opportunity for pop PK and more reliance on the early phase 1 days. I used to run many phase 1 research sites for usually human abuse liability and other types of studies. So incredible insights. Thank you. Thank you so much. And this is a perfect time, right, Sam, to transition to virtual networking and a longer break before the afternoon sessions. Is that right?
Yeah, we're going to add 10 minutes. So I guess we can delay yours and Laura's presentation by 10 minutes, if that's fine. And then before I do the transition, there's just one question for Dr. Nancy from Ronald, I believe, Senior Director of Supply Chain. He's asking, are the pharmacology requirements the same for cell therapy?
That's a great question. And it's interesting. I was going to put a little bit about that with the stem cell and the gene therapy. There's not the very specific bioanalytical approaches because of those types of treatment. But I will tell you, the FDA has now made it very clear that there are other parameters that they do want you to be looking at in lieu of the traditional PK, because they believe that will allow them the opportunity to assess the absorption distribution of these novel therapies, the gene therapy and the cell therapies. So they will, for example, expect us to do viral load testing. They will look for viral excretion. In the cell therapies, they do believe that and do require some types of immunogenicity and antibody testing because that allows them to do the safety assessments. And so it allows them, they really have some experts now working at this and looking at how over time you can do those types of assessments in lieu of the traditional ELISA or HPLC method of testing a smaller large molecule.
Perfect. Wonderful. Thank you so much. And as Dr. Nancy mentioned, if you have any other questions for her and her group, you can reach out to her via email. And yes, your slides will be available if anyone wants a copy. And yeah, we will be heading to our one of our breaks for the afternoon. And before then, I'll do one quick draw. We're doing these draws for the Amazon Uber Eats gift cards. Later today, towards the end of the summit, we'll do the raffle pick for our Apple Watch winner. But right now, I'll pick one winner for $200 Amazon gift card. So those that visited the booths thus far in the program, we have put their names here. So I'm just going to randomly, you know what I'll do? I'll put them all in my hat. And we'll pick one lucky winner. And right now, we have Miss Stephanie Calais from BMS, who have won $200 Amazon gift card. So feel free to email us, myself, Sherry, or Victor, and we'll help redeem that for you. Again, Stephanie Calais, if you're in the room, we'll also email you as well for the $200 Amazon gift card. We still have many more prizes, Uber Eats gift cards. Like I mentioned, at the end of the day, the Apple Watch draw, that will be happening. So please visit the booths during this hour break. Thank you very much. We'll connect again at 1:15 p.m.